Hello, my name is Trevor Frisbee. I am entering my fourth year as a graduate student in the Computational Biology Department at Carnegie Mellon University, and I am advised by Christopher Langmead. It is my great pleasure to meet with you all virtually, and today I am excited to talk about my most recent work, titled Bold Family-Regularized Bayesian Optimization for Directed Protein Evolution. I'd like to start with a concept that everyone here is well acquainted with, the central dogma of molecular biology. Informally, we describe this principle as DNA goes to RNA, which goes to protein. While this is obviously a vast oversimplification, what we are describing here is the way that biological sequences encode and pass information across different biological scales. While we can understand and explain evolutionary processes at any of these scales, this talk will focus on amino acid sequences and proteins. In particular, we will focus on the relationship between protein fitness and sequence. Depicted in the bottom image is the protein fitness landscape, which illustrates the relationship between protein functionality and its amino acid sequence. It is a complicated surface, with isolated regions of heavy undulation, often separated by deserts of non-functional sequence. Evolution and natural selection act by traversing these landscapes and do a pretty good job at identifying useful and functional sequences. However, this natural process can take extraordinarily long times. Additionally, the space of possible protein sequences grows exceptionally large, as does a protein's length. No natural or artificial process could ever evaluate them all, and good sequences are exceedingly rare. This leaves the question of how can we find these high-fitness sequences? Protein design and engineering tries to tackle this needle-in-the-haystack problem to identify sequences with novel or improved functionality. While this can include attempts to build proteins from scratch, more often than not, we use evolution's work as a scaffold. There are two primary methods used for designing proteins. While these approaches can be complementary, they tackle the problem of searching the protein fitness landscape in fundamentally different ways. Rational design uses computational and physical models to ascertain relationships between many properties of a given protein. Consider performing homology modeling as a typical example. This leads to hypotheses about the utility and functionality of novel or altered protein sequences. The specific sequences identified that are consistent with applied models are then synthesized in lab, where the properties of interest are then measured. In other words, a particular sequence is rationally crafted from first principles, and only then is created. These methods often have low throughput and can be limited by very costly computations. Alternatively, directed evolution is an approach that is traditionally model-free and uses wet lab methods to accelerate natural evolution and selection. These approaches typically rely on iterative mutagenesis techniques to create a library of variant sequences, followed by screening to identify those with desired traits, then finally amplification of these selected sequences. It is a much more guess-and-check approach to identifying improved protein sequences, but is a method that has proven very effective. Frances Arnold won the 2018 Nobel Prize in Chemistry for her work with directed evolution. Two examples of traditional directed evolution approaches are single mutation walks and recombination. Diagrammed here is the single mutation walk. This is a greedy procedure that evaluates all amino acid variants at a single position and fixes in place the best before continuing at other positions. Once the specific positions are chosen, randomly or otherwise, it is a deterministic walk across the fitness landscape through single site mutations. Given the exponential increase in number of possible mutations as we increase the number of sites mutated, this procedure can really only be employed at a handful of loci within a protein sequence. In a general setting, we may not know which positions would be best to mutate, making this a relatively inefficient way of identifying desirable sequences. Recombination creates new sequences by combining components of two or more distinct ones. This approach is intended to mimic natural genetic recombination that occurs via crossover events during eukaryotic meiosis. While this technique is capable of uncovering a wider array of variant amino acid sequences, it is still limited in scope, similarly to that of the single mutation walk. Although directed evolution has traditionally been a model-free approach, it can be well posed as a machine learning problem. Of course, machine learning refers to any number of statistical procedures that are capable of learning patterns from data, which can then be used to make predictions on unseen data. Here, we want to learn some function that maps protein sequence to its fitness. Having such a function would help to alleviate the biggest burden with respect to directed evolution, the cost and total amount of necessary experimentation. By their nature, traditional approaches require large numbers of protein syntheses. Finding ways to reduce the number of these experiments necessary before identifying high-fitness proteins would thus be hugely beneficial. To this end, we incorporate an active learning framework into the general directed evolution approach. Active learning is an iterative procedure where we learn a model on a small training set, use the model to assess which unseen data points would be most beneficial to obtain, then observe these data points and update the model accordingly. Essentially, we sequentially select new batches of training data, where each batch is tailor-made to best identify optimal data points. This procedure thus aligns well to a typical directed evolution approach, where sequences chosen in one batch serve as seeds for the next. This leaves the question of what model to use. For that, we turn to Bayesian optimization and use Gaussian process regression. Bayesian optimization is used to optimize costly or hard-to-compute functions. Aside from the actual cost of running experiments to determine a sequence's fitness, the protein fitness landscape is a very complicated, non-convex surface. We want to find a function that traverses this landscape from areas of low fitness to its peaks, like traveling from points 1 to 2 in this figure. To do this, we want to calculate the posterior distribution over protein fitness, conditioned on its sequence. An approach that has proven effective in similar and dissimilar domains is Gaussian process regression. At a high level, a Gaussian process generalizes the idea of fitting a Gaussian distribution to data to fitting a probability distribution over functions in n-dimensional space. It is fully defined by a mean function and covariance function, and allows for uncertainty calculations. The figure to the left shows a simplified example. Here, the 3 black dots represent sequences whose true fitness is known and used by the model. They lie on some true unknown fitness profile, represented by the black curve. The blue line is the most probable function under the Gaussian process, and the blue shading shows the model uncertainty. Notice how near the known data points the model closely resembles the true function, and does so with little uncertainty, while far from these points the model strays far from the true function value, with high uncertainty. In order to select sequences whose fitness will be measured, the posterior obtained from the Gaussian process is then passed to an acquisition function. We use expected improvement, which identifies the sequence expected to most improve upon the current optimal fitness value. This function may consider the Gaussian process's uncertainty, which can be a source that drives exploration of the sequence space versus exploitation of the current model. The bottom left figure shows an example acquisition function. We can simply identify the sequence that maximizes this function, denoted here by sequence K. After measuring the true fitness value of sequence K, We would update the Gaussian process in the next iteration accordingly to include this true fitness value. Notice how the updated Gaussian process now has less uncertainty, and how the selected sequences generally approach local optima of the true fitness value function. Lastly, we have incorporated into the acquisition function what we call fold family regularization. Protein fold families refer to groups of proteins that share homology and function. Proteins within a given fold family have been optimized through natural evolution to ensure that they have a full range of complex properties to function properly in a similarly complex cellular environment. Therefore, we should build off of what evolution has provided to guide our search of sequence space. To attain this, we use two different models, Gremlin and PFAM-HMMs, that both encode underlying statistical dependencies between sequences within a fold family. The Gremlin algorithm uses a Markov random field, and PFAM a hidden Markov model. While we omit specifics of these models, the primary difference between them is that whereas HMMs only encode dependencies between adjacent residues, the Gremlin algorithm can detect and encode both sequential and long-range dependencies. Either way, the models encode a joint distribution over residue types at each position in the primary sequence, which can be used to compute the log odds of given designs. We use this score to scale each sequence's expected improvement. Fold family regularization is thus a means of biasing variant selection towards those with native-like properties. We tested our Bayesian active learning approach using a 2016 eLife dataset that focuses on 4 positions of the GB1 protein, an antibody binding protein found on the cell surface of streptococcal bacteria. These positions are highlighted in the diagram below and were chosen by the initial study because they interact with each other in 3-dimensional space and are known to contain 12 of the top 20 positively epistatic interactions among all pairwise interactions in protein GB1. For our purposes, this dataset marks an extensive continuous portion of the protein fitness landscape. Stability and antibody binding affinity of about 149,000 GB1 variants were used to compute fitness values. These fitness values were normalized to a 0 to 9 scale, where the wild-type sequence has fitness 1. The total number of sequences obtained spans 95% of all possible variant sequences across the 4 sites studied. Only about 2.4% of all sequences measured had fitness greater than wild type. In our experiments, we used 3 different Bayesian active learning models, where GP plus EI refers to a Gaussian process with vanilla expected improvement acquisition, and the Gremlin and HMM models correspond to respective fold family regulated acquisition. In addition to these models, we also simulated the traditional single mutant walk and recombination procedures representative of traditional directed evolution techniques. We performed 10 iterative rounds of active learning and repeated this procedure 100 times, starting from different initial training sequences each time. Each experiment was designed to observe 190 total sequences, roughly equivalent to the experimental load of using 2 96 well plates. We find that Bayesian active learning is able to outperform simulated traditional directed evolution approaches. This figure shows the maximum fitness of a sequence selected by each method as a cumulative fraction of all 100 replicate experiments. As an example, at a cumulative fraction of 0.2, the blue curve, which corresponds to a standard Gaussian process with vanilla expected improvement, has a maximum fitness of just under 6. This means that 80 of the 100 trials selected a sequence that had fitness of about 6 or higher. In fact, this trend is true for each of the Bayesian active learning approaches. With the traditional approaches, fewer than half of the trials were as successful. If we look at the average maximum fitness values identified by each approach across all conducted experiments, standard GP plus EI finds the most fit variance and improves upon the fold family regularized approaches by about 7%. This is not entirely unexpected, as the fold family regularized models essentially have an additional constraint that selected sequences should share statistical similarity to sequences seen in nature. Still, all of the Bayesian approaches do better than a competing method from a 2019 PNAS paper by Wu et al. That trained shallow neural nets on the same data and even used a larger training set. We emphasize that their approach did not use an active learning setup. Bayesian active learning approaches can thus improve directed evolution. While fold family regularization induces a small penalty in fitness of selected sequences, they are more than compensated by the gain in log odds score under a generative model for the GB1 fold family. The left figure shows the per batch fitness of sequences selected by each model. While GP plus EI identifies sequences with higher fitness, it only does so by a thin margin over the fold family regularized models. The right figure shows gremlin log odds scores of selected sequences and demonstrates that the regularized models select sequences that are upwards of 40% more native-like than the unregularized model. Such sequences thus, on average, share more statistical properties with sequences seen in nature, while also, on average, improving fitness. These sequences could be more likely to contain beneficial properties that could be evident in nature that may not be captured fully by a fitness assay. Finally, we use the models we learned to make fitness predictions on a held-out test set of 30,000 GB1 sequences. Recalling that the fitness values lie on a 0 to 9 scale, we find that these models generally tend to have low accuracy, where mean squared error tends to be greater than 1. Wu et al. Report a similar result with their neural net models using the same dataset. Interestingly, this means that we can make headway towards identifying high fitness sequences without needing to precisely map sequence to fitness. We also observed that properties induced by fold family regularization carries over into predictions made on the held-out test data. At the end of the 10-batch active learning cycle for each model we learned, we performed one final sequence selection step over the entire 30,000 held-out test sequences. We then identified the top 100 scoring selected sequences according to each model, and in these figures, we report the true scores of these selected sequences. In the left figure are the distributions of true fitness scores. As we saw before, we noticed that while both the regularized and unregularized models are able to select high fitness sequences, the regularized models are slightly penalized in terms of fitness of their selected sequences. In the right figure, we show the true gremlin scores of these same selected sequences. Here, the regularized models do a better job at identifying sequences that are evolutionarily more similar to those in nature. Thus, fold family regularization can induce a trade-off between the nativeness of novel sequence designs and that of some experimental property of the protein, such as fitness. To summarize, directed evolution is a traditionally model-free approach to finding improved protein sequences by accelerating natural evolution and selection. We have shown how directed evolution can be enhanced through active learning. More specifically, we have applied Bayesian optimization and demonstrate how it can improve upon traditional and other machine learning-based directed evolution strategies using data from the GB1 protein fold family. By using fold family regularization, we can search through protein sequence space to identify sequences that improve fitness while sharing statistical properties with existing sequences in nature. Finally, while the models we learn tend to have low accuracy, they maintain any selection biases that were induced by fold family regularization. I'd like to acknowledge that this work is funded by a T32 training grant through the HHMI NIBIB Interfaces Initiatives. With that, I conclude my talk I would now be happy to take any questions.